Welcome to the C-Suite Series presented by Channelchek and Noble Capital Markets. Noble is an SEC-registered, FINRA-licensed broker-dealer and the source of the equity research available on Channelchek. Of the 6,000 small and microcap companies listed on the site, today we're featuring PDS Biotechnology. Ahu Demir, Noble's research analyst covering the company, will be interviewing CEO Frank Bedu-Addo. Afterwards, she will be joined by Michael Kopinski, Noble's Director of Research, to discuss the key takeaways. As a reminder, Ahu's research is available at no cost on Channelchek. Just type in PDS Biotechnology in the top left Company Data section. And now, here's Ahu and Frank.
Thank you. Here is the CEO of PDS Biotech, Dr. Frank Bedu-Addo. Thank you so much for joining us today, Frank. We look forward to speaking with you. Thanks for your time.
Thank you very much for having me.
So I'll start with my first question. If you can provide us some introduction of yourself, your background, and also quick overview of PDS Biotech's current portfolio.
Well, thank you very much. I'll start with my background. So for the last several years, I've been in senior management and executive roles overseeing operations at drug development companies such as Cardinal Health and KBI Biopharma, where I was one of the founding executives. In my earlier career, I was a scientist and have been involved in the development and manufacture of several pharmaceutical products at companies such as AkzoNobel and Schering-Plough, which is now Merck. And so going to the second part of your question regarding our portfolio, PDS Biotechnology has quite a deep and broad product pipeline. We focused on two key areas. First, our oncology programs. So currently, our oncology pipeline consists of 4 products, which address approximately 10 different types of cancer, with our lead program being PDS-0101, which addresses various HPV-associated cancers. And our lead program, this lead program is currently undergoing phase 2 human clinical trials. We also have our infectious diseases pipeline, and in this case, we have 4 products currently in preclinical development, and our lead program is the development of a second-generation COVID-19 vaccine.
Thanks, Frank. I think I will start with the platform technology prior to talking about the pipeline assets. Since your portfolio is built on Versamune platform, why do you believe it has such a promise in immunotherapy?
Well, the promise of the Versamune platform technology really lies in the immunological characteristics and attributes of this novel technology. So Versamune has been engineered to be simple and efficient. And the way our products work with Versamune is, with most cancers or several cancers, these cancers have certain unique proteins that are either displayed or contained within these tumors that can be recognized by our immune system. And these proteins are not highly expressed or non-existent in our normal healthy cells. And so what we do is we take that protein that's unique to the specific disease we're seeking to treat, and we combine it with the Versamune platform technology. Now, what the Versamune platform technology does is quite important. First, it's able to take this protein and deliver it into the right immunological pathway that allows it to be effectively processed and broken down by the immune system. And then that processed protein then presented into an important pathway known as the MHC class 1 presentation pathway. This allows that processed protein to be presented to an important subset of our T cells, which are attackers, known as CD8 or killer T cells. And so this process of presenting that processed protein to the killer T cells is what trains or primes these killer T cells to specifically recognize the cancer. Now, this has been one of the significant limitations impacting immune oncology today, specifically in vivo priming and activation of these tumor-recognizing killer T cells in humans. The second thing our technology does is it activates a novel pathway known as the type 1 interferon pathway. Now, activation of this pathway is important for these trained T cells to be effectively activated and to enhance their killing activity, and also to enable effective activation of multiple large numbers of these tumor-attacking T cells. And so by being able to perform these critical functions of priming and activating or training these killer T cells, as well as activating them, and also overcoming the tumor's immunosuppressive mechanisms, allows for an effective immunotherapy. And by performing these functions, as I mentioned, overcomes one of the major limitations facing immuno-oncology today. These novel mechanisms were recently published in one of the top peer-reviewed journals, which is the Journal of Immunology, in June of 2019, really covering how the Versamune technology is able to perform these critical functions related to anti-tumor immune responses.
So my next question would be then your lead candidate, PDS-0101, targeting HPV-associated cancers. So what is the current status of your phase 3 clinical trial for PDS-0101? And could you also highlight the differentiating factors in the competitive landscape?
Sure, I'll start with the first part. So with PDS-0101, we currently have 3 phase 2 clinical trials planned. The first phase 2 clinical trial is already in progress. This started in June of this year, and this is led by the National Cancer Institute. This is a trial that addresses advanced HPV-associated cancers. This is a novel triple combination that combines our PDS-0101 with M7824, which is a bifunctional checkpoint inhibitor owned by EMD Serono, as well as NHS-IL12, which is an immunocytokine also owned by EMD Serono. This clinical trial was based on preclinical work that was done by the National Cancer Institute, demonstrating a strong synergy between these 3 compounds, leading to dramatically enhanced anti-tumor effects. And this preclinical study was published by the National Cancer Institute in the Journal for Immunotherapy of Cancer last month. So this trial, as I mentioned, is currently underway and recruiting according to schedule. The second clinical trial is led by Dr. Anne Klopp of the MD Anderson Cancer Center. And this trial combines PDS-0101 with the standard of care chemoradiotherapy. This is focused on advanced localized cervical cancer. And the goal here is, in this combination of PDS-0101 and chemoradiotherapy, being to enhance the clinical benefit to these patients over just the chemoradiotherapy on its own. This trial we anticipate will get started in the next few weeks. The third trial is a collaboration with Merck, and this combines PDS-0101 with Keytruda, a checkpoint inhibitor, to address first-line treatment of recurrent or metastatic head and neck cancer. This trial, PDS announced in June that we were putting this trial on hold due to the COVID-19 pandemic. We've been in contact with the sites that were qualified to run the trial, and we do anticipate initiating this trial before the end of the calendar year. So those are the three phase 2 clinical trials for PDS-0101 that we currently have initiated or will be initiating very soon. Now, in terms of the differentiating factors, HPV cancer immunotherapy is quite an active space. There are a number of companies developing products to address these cancers. We're looking at different kinds of immunotherapy. We have live vector, live virus, and live bacteria-based, DNA-based immunotherapies. We have peptide adjuvant-based immunotherapies. We also have combinations with checkpoint inhibitors, to mention a few. Now, with PDS-0101, one thing to bear in mind in terms of how this is differentiated is, with a PDS-0101 monotherapy, we have demonstrated that the monotherapy on its own overcomes significant limitations of immuno-oncology today. So, for example, the ability to activate these tumor-targeting killer CD8 T cells in vivo, and subsequently leading to regression of lesions in vivo in humans as a monotherapy. Our strategy then has been, as you can see in 2 of our trials, to combine with the standard of care. And so with the Merck trial, for example, we're combining with Keytruda, which is the approved first-line treatment standard of care for recurrent or metastatic head and neck cancer, with the goal being to take this product that has shown efficacy as a monotherapy, combine it with the standard of care to enhance the clinical benefit to the patients. We've taken the same approach with advanced localized cervical cancer, where we're combining PDS-0101 with the FDA-approved standard of care, which is chemoradiotherapy. Now, I also talked about the novel triple combination that we looking at combining PDS-0101 with M7824 and NHS-IL12. So if we are able to replicate the highly promising preclinical results in humans, this would be the basis for a novel triple combination platform that can then be broadly applied to a number of solid tumors. And so these are very important differentiators regarding how we are approaching addressing these HPV cancers. The Merck trial is actually quite important to really emphasize the fact that we are addressing first-line treatment of these patients. Right, so Merck is really has the first immunotherapy Keytruda approved for first-line treatment of patients using an immunotherapy. And so these patients will be able to go on the combination before they're even treated with chemotherapy.
Thanks for that detailed explanation, Frank. So my next question will be, what is in the oncology pipeline beyond PDS-0101?
So PDS has 3 additional products being developed in the oncology pipeline. The next product after PDS-01 is PDS-0102 and PDS-0103. Both of these, with both of these products, formulation development is already complete. PDS-0102 combines the Versamune platform with a novel protein discovered by the National Cancer Institute to be present in approximately 90% of prostate cancers and 50% of breast cancers. And so PDS-0102 specifically addresses prostate cancer and breast cancer. The National Cancer Institute has performed a clinical trial where they took this protein and studied it in advanced prostate cancer patients and confirmed that the human immune system recognizes this tumor-associated protein, and this led to a dramatically reduced rate of tumor growth progression in these prostate cancer patients. What PDS has demonstrated is that if we take that same protein and combine it with our Versamune platform, what we see is a dramatically enhanced anti-tumor T-cell response generated simply by combining it with Versamune based upon the much more effective presentation to the immune system as well as activation of these killer T-cell populations. So we believe that PDS-0102 could potentially provide a suitable therapy or an effective therapy for both prostate and breast cancers. Now moving on to PDS-0103. PDS-0103 addresses 4 cancers, again, breast cancer, colon cancer, lung, and ovarian cancers. And in PDS-0103, again, we are taking a protein that's expressed on multiple cancers, each of these 4 cancers I mentioned. Again, the National Cancer Institute has developed a novel analog of this protein that is much more readily or much more strongly recognized by the human immune system. So again, we're taking these and combining these with the Versamune platform to generate a much more powerful anti-tumor effect. We recently announced that we have expanded our collaboration with the National Cancer Institute to now include development of PDS-0103. And so what that entails is the National Cancer Institute will be performing preclinical studies and also evaluating combinations with other immunotherapeutic agents, and based upon those results, potentially progressing it into a phase 2 human clinical trial. And so we are hopeful that this will be progressed into human clinical trials sometime next year. PDS-0104 is the other product currently in development, which addresses melanoma. And this is in the earliest stage of development compared to the 3 other products.
Thanks, Frank. So that shows the richness of the platform technology that you have, that you can use multiple, you can drive multiple assets in many different cancer indications. So if you would move from oncology to infectious disease, you recently announced an expansion of your pipeline to infectious disease. And could you please tell us what data have you generated with Versamune in infectious disease, and could it potentially address the need for COVID-19 pandemic as well?
Yes, we believe there's strong potential for the Versamune-based vaccines in COVID, and I'll get to that. With the infectious diseases, we've taken a very similar approach to what we've done with oncology, essentially partnering with some of the top players in the field, which has allowed us to have that breadth, right, in terms of developing multiple products in multiple indications via these partnerships with leaders in the field. A very similar approach with our infectious disease programs and portfolio. So in infectious diseases, we currently have 4 products in development. The first product, PDS-0201, is addressing tuberculosis, which today remains a significant world health issue. And this is being developed in collaboration with our partner Farmacore in Brazil. And this is based upon a novel TB protein that has been developed by Farmacore and is being combined with the Versamune technology. The initial results have been very promising, and we're continuing development of this product. The second one is PDS-0202, which addresses influenza. So this would be what we would call a universal flu vaccine. And by universal, I mean that this vaccine will be applicable to multiple strains of the flu virus. And we have done our initial preclinical data has demonstrated rapid and powerful induction of both neutralizing antibodies against flu, as well as T-cell responses against flu. And so based upon these early results, we also recently announced a grant award by the NIAID to one of our collaborators, Professor Gerald Woodward at the University of Kentucky School of Medicine, to complete preclinical development of this Versamune-based universal flu vaccine. And so here also we're quite hopeful that we will be able to progress this into human clinical trials sometime next year. And then we move over to our COVID-19 vaccine. Our COVID-19 vaccine has been informed by quite a bit of the knowledge that has been gained due to this recent pandemic, right? So based upon the emergent data, we know that in addition to neutralizing antibodies, our immune system actually generates T-cell responses against the virus. And the emerging data suggests very strongly that a much broader range of immune responses would be necessary by a vaccine, an effective vaccine, to effectively prevent spread of the virus, as well as to prevent a durable long-term protective effect against the virus. And based upon this knowledge, as you can see, PDS is very well positioned with our Versamune technology, which is really geared towards generating these powerful T-cell responses in vivo in humans. And based upon that mechanism of action of this technology, what we decided to do was to rapidly evaluate its potential in development of a COVID-19 vaccine. I referred to this as a second-generation vaccine. And the reason why we call it a second-generation vaccine is just differentiated from what's currently being done, which is a real emphasis and focus on the induction of neutralizing antibodies. Based on the data that's been generated today in the field and the emerging knowledge of the breadth of T-cell responses required, what we are focusing on is not only generating those antibody responses, neutralizing antibodies, but the right type of T-cell responses that will be required to produce that robust anti-COVID-19 response. So looking at killer T-cell induction, which has been shown to be important, looking at helper CD4 T-cell induction, which is also shown to be important, and very importantly, memory T-cell induction, which will provide that long-term protection against the virus. And as we know, not all T cells are created equal. So the other thing we've been doing is looking at the actual potency of the T cells that we're generating to ensure that these are what we would call polyfunctional multi-cytokine inducing T cells, which are the most powerful type of T cell. And so to date, our preclinical data has actually confirmed that we are able to induce this breadth of immune responses against COVID-19. So PDS-0203, for example, contains a protein, a recombinant protein. So this is what we call a subunit vaccine. So it's based on recombinant proteins rather than live viruses, RNA or DNA. So we take that protein from the SARS-CoV-2 virus, a portion of it that the antibodies are able to respond to, and it also contains a region of the virus that our T cells can also recognize and respond to. And so what we have demonstrated is that within 14 days of vaccination, we're able to generate a powerful neutralizing antibody response. Approximately over 90% of the antibodies are shown to have the neutralizing effect. And what's also very important and unique is the fact that after that vaccination, even without further vaccination, over a 30-day period, we continue to increase the antibody levels in these preclinical animal models. And this is based on the mechanism of action of Versamune, where we get a prolonged and continued priming of these antibodies and T cells. We show a very similar effect with the T cell responses, where within 14 days, we can see about a 35 to 45-fold increase over the non-Versamune containing vaccine, and also demonstrating these strong memory T cell responses even after 60 days after vaccination. We believe that a second generation vaccine should have all these attributes in terms of both the antibodies and strong and powerful T cell responses. It should also be safe. It should be easy to scale up and manufacture at a commercial scale. And it should also be stable and facilitate transportation across the globe. All these are attributes that are exhibited by the Versamune-based vaccines. And so we've presented our initial preclinical studies to the FDA and already received some initial feedback from the FDA in terms of the final preclinical studies and the clinical strategy for PDS-0203. PDS-0204 is also a COVID-19 vaccine. However, in this case, it is being developed in partnership with the Brazilian company Farmacore. So it's based on a protein that has been designed by Farmacore and is combined with our Versamune platform. It is anticipated that the funding for this program would potentially come from the Brazilian government. The initial funding that covers the preclinical studies has already been received by Farmacore, and the goal would be to complete these preclinical studies hopefully by the end of this calendar year and move that program into human clinical trials either at the end of this year or very early next year.
Thank you, Frank. That was great detailed information again. So we appreciate that. So let's switch gears. That I was curious how much cash you have in hand. What is your current cash burn, and also how do you prioritize, considering you have such an enriched pipeline? How do you prioritize your capital use between oncology and infectious disease?
Well, interesting question. So we recently announced that we completed a $19 million financing. And based upon that, with the cash we previously had in hand, the company has a little over $30 million in cash currently. Now, if we perform the planned phase 2 human clinical trials for PDS-01, assuming we perform all those trials, we would have enough cash to take the company approximately 2 years. So we have about a 24-month runway with the capital that we've raised. Now, in terms of how we prioritize that cash, so we would want to continue our approach of partnering with some of the leaders in the field to build out our pipeline. The goal is really to prioritize this funds we've raised to our oncology programs and to build out those oncology programs and further them through rapid proof of concept in these larger phase 2 clinical trials. With infectious disease programs, our priority right now is to potentially continue to partner, seek these partnerships to move forward, as well as potentially government funding, just like we've done with the universal flu, and also what we would hope to do with our with the PDS-0201. But with the infectious disease programs, currently the goal is to seek either government or non-governmental organization funding, and also partnerships to move those into clinical development, while we focus the funds we've raised on the oncology programs.
Thanks for that information, Frank. So my last question will be, what will be the value-generating catalyst to keep an eye on for the next 12 months?
So let's start with the oncology programs. So with the oncology programs, I think one of the key things we want to accomplish in the next few weeks is get the MD Anderson trial in advanced cervical cancer going. This was the combination with the standard of care radiochemotherapy. That trial hopefully will get going in the next month or so. Secondly, we would also want to get the study with Merck, combination PDS-0101 with Keytruda. That's the trial that was put on hold due to the COVID-19 pandemic. the goal is to get this trial up and running before the end of the calendar year. So that's another key milestone that we will be achieving. And with the trial that has already started, in the triple combination, the hope is to start to generate some data, which hopefully the NCI, this is an NCI-led trial, and we are hoping that they will have some initial efficacy data to make public probably sometime late in the first quarter of next year. With the infectious disease programs, we are hoping that we will be able to get one of these, the PDS-0203 or the collaboration with Pharmacore 0204, at least one of those into human clinical trials, either before the end of the calendar year or early next year. So those are all key milestones that we are working towards and anticipate hopefully being able to announce in the very near future.
Thank you, Frank. We really appreciate your time and all the explanations. Thank you so much for joining us today.
Thank you very much.
Ahu, I'm always fascinated by, treatments of cancers and things like that. I mean, I can't think of anyone who has not been affected by someone that has had cancer, or especially even the COVID situation, right? So I'm interested in, Frank said that this is a novel approach. Can you talk a little bit about the triggers with the T cells and the way it's delivered? It seems quite interesting to me and different than what is out there. And I was just wondering if you can explain or just expand upon that a little bit more.
Yeah, of course, Michael. So the delivery system they are using, it's a Versamune, which is a T-cell activating platform technology, and it is designed to instruct the immune system to recognize and attack cancer cells or infectious disease. So the Versamune technology is a tumor-specific T-cell response that generated, and there are two components of it. The first one is structure-specific positively charged lipids, and the second part is the custom design antigens targeting different types of cancers or infectious disease. So it is delivered via simple subcutaneous injections, and it's a nanoparticle. It binds to cell surface based on the positively charged cationic lipids, and it's uptaken by the cells. So it's delivered to the lymph nodes and stimulates, present the antigens via dendritic cells, and it triggers the T-cell responses. So it has a very unique platform technology based on these facts.
And Frank stated, just kind of moving on to the COVID situation, because it's top of mind, obviously, he stated that he's looking at a vaccine that's more of a second generation vaccine. Is that because of the complication of the novel way he's planning to neutralize the antibodies, or is it the length of time it will take for clinical trials, or maybe both? Can you explain why it would take so long?
I would say it's both. The timing definitely affects. It's going to take some time to advance it into the clinic. And also, as Frank pointed out, majority of the current vaccines are focused on neutralizing antibodies, and Versamune technology will be finding and recognizing the antigen, presenting it to the T cells, and generating active killer T cell responses. So both of this timing and the uniqueness of the technology would be the reason to make it a second generation vaccine.
Well, thanks for that clarification, Ahu. I really appreciate it. It's a complicated subject, but you made it simple, and I appreciate that. Well, it was a great interview. Thank you for bringing the management.
My pleasure. Thanks for your questions, Michael.